A wide variety of compounds can be included as a carbon source in an industrial fermentation media. And the type of carbon source will always depending on the type of microorganism and type of process. In this video, I am going to talk about properties of some of the most commonly used carbon sources in industrial media. In some cases, we use these carbon sources in combination with other components. Let us see what are the properties of commonly used carbon sources. One of the most commonly used carbon sources is molasses. Molasses is a byproduct of sugar industry. When we are making sugar from cane or beet, there is a process of crystallization. The juice from this cane or beet will be concentrated and go for crystallization. That's how we get the sugar crystals. And after crystallization, the compound like residues we get after crystallization is called molasses. Molasses is a dark viscous fluid. As you can see in the figure, it is a very dark viscous compound. It's actually a waste product of sugar industry. So it is cheap and it is widely available. And it is being used for the fermentation of many different components. If you look at the composition of this molasses, 50 to 60% of molasses is composed of carbohydrates, and basically it is sucrose. And it has 2% nitrogenous compounds like proteins, amino acids, and other compounds. It also contains some vitamins and minerals. The exact composition of the molasses is depending on very different factors, which include the plant source, whether we are using a cane or a beet, and what the type of crop we are used, and what's the location of crop, in how much irrigation it is getting, what are the climatic conditions, and the type of factory it is using, what is the process of crystallization. So the exact composition will vary very widely depending on the type of plant and type of industry. And it basically, the molasses is used for the production of low value high volume products. So as we know, low value high volume products, uh, in products including this ethanol, organic acid, and single cell protein. In the production of these low value products, there is a huge impact of media components. If you are using waste product as a media, it will be more economical. So molasses is a waste product of an industry, so it is economical to use in low value products. It is also used for uh, fermentation of some of the high value products such as enzymes, but for the production of secondary metabolites like antibiotics, we don't really use this molasses as a substrate. The reason is that molasses contain mostly sugars like monosaccharides, readily available sucrose. Uh, so, not monosaccharides, actually sucrose. So, as I told in previous video, the readily utilizable sugars will inhibit the production of secondary metabolites. But this molasses can be used for the production of primary metabolites like ethanol, organic acids, or the biomass and enzymes. One other factor is that if you store molasses for a long time, its sugar concentration will reduce because of the contamination. It is not a pure carbon source. It's a waste product, so it has so many impurities. So we cannot store it for a long time. If it's stored, the microorganism will spoil it and the sugar concentration will reduce. One of the related molasses is called hydrol molasses. Instead of producing from the sugar cane or the beet, the hydrol molasses is a byproduct of maize starch processing industry, and it also primarily containing glucose as a main component. Next carbon source is malt extract. It's an aqueous extract of malted barley, which is concentrated to form malt syrup. So it's a originated from malt. So I guess you know how to the barley is used in different industries, and you also know the process of malting. It is actually germinated to get the enzymes, and after the germination, we will take it and it is made at a paste. Basically, it is used for the production of beer, and it is a good carbon source for the cultivation of filamentous fungi, yeast, and actinomyces because it contain the some of the complex carbohydrates like polysaccharides. If you look at the composition, it contains 90% carbohydrates and 5% nitrogenous substance like protein, peptides, and amino acids. So it's a very good carbon source and it's an average nitrogen source. Microorganisms do not need huge amount of nitrogen when compared to carbon. So 5% nitrogen is not bad. Next carbon source is starch or dextrins. Starch or dextrins are basically polysaccharides, so it is not readily utilizable. So it can be used for the production of secondary metabolites. So it can be directly used for the growth of filamentous fungi, producing amylase, filamentous fungi. The thing is that if a microorganism need to use the starch as a substrate, it should have a mechanism to degrade these polysaccharides into monosaccharides. So the only the organism which has the ability to produce amylase, which is the enzyme degrading starch into sugars, can use starch as a carbon source. We cannot use starch as a carbon source for Saccharomyces cerevisiae. Saccharomyces cerevisiae can only use monosaccharides and disaccharides. It cannot use starch as a carbon source. So starch is a polysaccharide. So only the microorganism which is able to produce amylase enzyme or some other enzyme which is degrading starch converting into monosaccharide can use this starch as a carbon source. And most widely used starch is maize starch. uh, in some cases, instead of using starch directly, what we take the starch as a carbon source, then we will convert the starch into sugar. And it is a conversion of starch into sugar is a two-step process. First we will go for gelatinization. After gelatinization, starch is degraded either using diluting acids or enzymes to get monosaccharides and disaccharides. And it is widely used for the production of glutamic acid. And for the production of glutamic acid, we are using hydrolyzed cassava starch. So when comparing starch with the molasses is basically containing monosaccharides and disaccharides. Maybe as it is a originator from the sugar industry, it must contain most of sucrose. But starch is basically containing polysaccharides. So it will be starch will be better for the production of antibiotics by the fungus, and the molasses will be better for the production of monosaccharides like primary metabolites like alcohol, organic acids, or uh, biomass. Next one is sulfate waste liquor. Sulfate waste liquor is also a waste product. It is originated from paper pulping industry. As the name indicate, it contains sulfate in it because they are using sulfuric acid, things like that, for the process of pulping. So the waste generated from the paper industry also contains the sulfate, and it has very low pH. That's why it's acidic in nature. As it is a waste product, it's very cheap. It is primarily used for the cultivation of yeast for the bio mass production and single cell protein production. And the characteristics of this sulfate waste liquor vary depending on the type of plant material used for pulping. If you are using coniferous trees for the pulping and paper making, it contains about 2 to 3% of sugar. Out of this 2 to 3 percentage, 80% are hexoses like glucose, mannose, and galactose, and 20% of them are pentoses. Pentose means the sugars with 5 carbons, and hexose is the sugars with 6 carbon. And as we know, hexoses are more common in biological systems. Pentoses are there, but it is not that common. But if the pulping is from the deciduous trees, which mainly contain pentoses rather than hexoses. So the sulfate waste liquor originated from coniferous trees containing more hexoses, but the sulfate waste liquor originated from deciduous trees containing more of pentoses. And one thing is that this sulfate waste liquor containing high amount of sulfite in it in the form of sulfur dioxide. So before using it as a carbon source, this sulfur dioxide must be removed. We can either go for a precipitation or this stream stripping to remove this sulfur dioxide so that the sulfur component will be removed. Another thing is that it has a very low pH, so sometimes we will use calcium hydroxide, things like that, to adjust the pH so that the microorganism can grow in that. And the nitrogen composition of the sulfate waste liquor is very low, So, and phosphorus is also low. So in order to use it as a medium, before using as a medium, we will add some nitrogen and phosphorus to it so that it will have more of nitrogen and phosphorus. So next is whey. Whey is also waste product. It is originated from the dairy industry. When we are making cheese out of milk, the liquid which came after the production of cheese is called whey. Annually, worldwide production of whey is about 80 million tons. So it's a huge amount. It is produced, and it is a waste product, and it contains good amount of carbohydrate and some amounts of protein. But the thing is that it is very expensive to store and transport. So what people do is that before storage and transportation, they will concentrate it. This way can be concentrated using evaporation, so the volume will be reduced. Once the volume is reduced, it is easy to transport and store. And sometimes people will, what they do is that they will remove the milk protein from the whey. So some of the milk protein will end up in the whey. So this milk protein can be used in different formulations and different types of foods. Maybe as a protein powder or something like that, we can use this whey proteins. Uh, the composition of whey is mainly contain milk sugar. Milk sugar is lactose. The thing is that lactose cannot be utilized by many of the bacteria. As we know, the lactose fermentation is unique to enterobacteria. So Escherichia coli and things, group of Escherichia coli, Shigella, things like that can use lactose, but uh, the use of this lactose is not universal. Only few microorganisms, enteric microorganisms can use lactose, but it is used for the production of some fungus like penicillin production. Lactose is not a readily utilizable sugar, so it can be used for the production of secondary metabolites. So, as an overview, the whey is a main product of dairy industry, and it contains protein and lactose. Protein can be separated for use as other purposes like protein powder or food formulations, etc. And lactose is say not really used by many bacteria, so it is only used by enterobacteria. In case of fungus, it can be used for the production of secondary metabolite. So these are the some of the important carbon sources. In next video, I'll talk about some rarely used components as carbon source. Thank you.